Next subtopic is components of flow cytometry. We will discuss one by one different components of flow cytometry. First of all, flow sheet. Flow sheet or sheath fluid is isotonic buffer which carries and aligns the cells to pass in a single file through the light beam for analysis. So along with cells or particles, we also use this flow sheet or sheath fluid, and this surrounds the sample, and this helps in isolation of the cells or passage of the cells through the instrument in a single file. Sheath fluids, like I mentioned earlier, surrounds a thin co-thread of sample which spaces the cells in such a way so that only one cell passes the laser beam at a time. These fluids or flow sheet that should be free of debris and air bubbles, otherwise analysis becomes difficult. And at the same time, this sheath fluid is properly pressurized at all the times so that it passes uninterrupted through the instrument. Now, optics which are used in this instrument, lasers and other optics, they're used for excitation of fluorophores, like I mentioned earlier. Excitation and emission of specific color of light depends on the type of fluorophore which are used. When laser light is used, these laser light, they produce two types of scattering, forward scattering, which is in front of the light beam, and the side scattering, which is on the sides. Forward scatter signal intensity is proportional to cell size, while side scatter signal intensity is proportional to granularity of the cells or properties. So with the help of forward scatter, we are able to measure the size of the cells, while with the side scatter, we are able to measure whether the cells or the particles are granular or not. This is shown here that on the left side, we can see a sample is being introduced into the instrument, and on both sides, on all sides of the sample, we have sheath fluid, which makes these cells pass one by one through the instrument. On the right side, in the red color, we can see laser beam, which excite the fluorophore, which are used to stain the cells, and then we have forward scattering and side scattering, with the help of which we are able to measure different characteristics of cells. On the right side is a dot plot. On the bottom of it is FSC, which means forward scattering. On the left side is SSC, like we earlier mentioned, is side scattering. So as we move to the right of the forward scattering, it means the cells become larger in size, while if you move further up, it means the cells are granular as compared with the lower cells. So, जो यहाँ right top पे population है, it has more size and they are more granular, while the cells which are at lower and on the left side, they are less in size and they are less granular. Neutrophils, which are found in the blood, they have higher side scattering than do agranular lymphocytes. This is what we mentioned, like in the previous diagram, that neutrophils, they are larger in size and they are granular as compared with lymphocytes, which are small in size and they are agranular as well. Dead cells, they have lower forward scatter and higher side scatter than the living cells. That's why they are dead, and when they're dead, their size, they shrink, they become granular as well. Photodiode collects forward scattering signals, and photomultiplier tubes collect emitted side scattering signals and fluorescent signals as well. Various mirrors and filters, they transmit these signals to their respective detectors. Data is presented in the form of a histogram if we are analyzing the cells on a single characteristic, and if we are characterizing the cells on a multi-parameter basis, then cytogram term is used. Now, electronics which we use in this instrument are able to monitor and control working of the flow cytometer. An electrical pulse is generated as cells or particles pass through the laser beam and in this way generate light signals. When the particle is in the center of the laser light, the strong signal is produced, and accordingly, when they are not in the center, weak signal is produced. And this signal is presented to the signal processing electronics and sent to the computer for further analysis. In the construction of histograms or cytograms like we earlier mentioned.